Hello everyone, my name is Alfred. I'm a graduate student at the University of Eldoret and also a practicing biomedical research scientist. So today we are starting our fascinating presentations on immunology, and this is the first upload, and I'm looking forward to discussing various aspects of immunology in our subsequent presentations. Hope someone finds this helpful. So let's begin. So in today's lecture we are going to cover introduction to immunology. So under this section we are going to define what is immunity. We shall also give the definition of immunology, the types of immunity, that is the innate and the adaptive. We shall also discuss the components of innate and adaptive immunity. Then we shall look at terms commonly used in immunology, and lastly we shall discuss briefly on organs of the immune system. So immunity is derived from the Latin word immunis, which means exempt or protect from a liability. And in medical science, immunity refers to being protected from infectious diseases or infectious pathogens. So we can define immunity as resistance to disease, specifically to infectious diseases, and it is the protection of the host from infectious agents. Moving on to immune system, so this is composed of the collection of cells, tissues and molecules that mediate resistance to infections. And immune response is the collective and coordinated response of cells and molecules of the immune system to infectious microorganisms or foreign substances. And lastly, immunology is the study of immune system, including its responses to microbes and damaged tissues and its role in disease. So the main function of the immune system is to prevent or limit infections by pathogenic microorganisms, for example bacteria, viruses, parasites and fungi. And it is important for you to also understand that the recognition of microorganisms and foreign substances is the first event in the immune response of a host. So next we shall discuss the types of immunity, and like I pointed out in our outline, we have two types of immunity: the innate immunity and acquired, which is also referred to as adaptive immunity. And both innate and adaptive immunity have two components, that is the humoral immunity and the cell-mediated immunity. So starting with innate immunity, so this refers to the mechanisms that are ready to react to infection even before they occur. So they are the first line of defense of our body against disease-causing agents. So it is also referred to as natural or native immunity. And this is the type of immunity that an individual usually possesses by birth. So what are the distinguishing features that distinguishes innate immunity from adaptive immunity? So number one, innate immunity is non-specific, meaning it can act on any pathogen. It is not specific to a pathogen or a disease-causing agent. Then number two, it acts within minutes. And next is that it does not improve after exposure, and it has no immunological memory. And innate immunity is classified into three categories. We have the individual immunity, racial immunity, and species immunity. So we are going to discuss this briefly one by one. So starting with individual immunity, this denotes resistance to infection, which varies within different individuals in the same race and species. And it is important for you to know that this type of immunity is genetically determined. For example, if one homozygous twin develops tuberculosis, there is a very high probability that the other twin will also develop tuberculosis. And in heterozygous twins, there is a very low possibility of the other twin suffering from tuberculosis. Moving on to racial immunity, this one denotes a difference in susceptibility or resistance to infection among different races within a same species. For example, races with sickle cell anemia are immune to infection caused by malaria parasite Plasmodium falciparum. And this is because of a genetically abnormal erythrocyte, which appears as sickle-shaped, and that sickle shape prevents parasitization of falciparum. Also, individuals with a hereditary deficiency of glucose 6-phosphate dehydrogenase are also less susceptible to infection by Plasmodium falciparum. Concerning species immunity, this one denotes a total or relative resistance to a pathogen shown by all members of a particular species. For example, chickens are resistant to Bacillus anthracis, whereas rats are resistant to Conibacterium diphtheriae. But these two microorganisms are susceptible to humans. And the exact reason for such type of immunity is still unknown. So next, we shall discuss factors influencing innate immunity. And we have three major factors which influence innate immunity. That is age, nutritional status of the host, and hormonal levels. So we shall also discuss this one by one briefly. So starting with age, it is important for you to know that extremes of age make an individual highly susceptible to various infections. And older individuals have waning immunity, whereas very young children have immature immune system. And for the fetus in utero, it is usually protected from maternal infection by the placental barrier. However, HIV virus, rubella virus, cytomegalovirus, and Toxoplasma gondii cross the placental barrier and cause congenital infections. So on that point of age, it is important for you to know that very old people are susceptible to suffer more than young people from a disease. For example, pneumonia, and they have high mortality rate. Diseases like measles, mumps, poliomyelitis, and chickenpox cause more severe clinical illness in adults than in young children. And this is because there can be more active immunity or more active immune response in an adult, which causes greater tissue damage compared to the children. Moving on to nutritional status, it is critical for you to know that both humoral and cell-mediated immunities are lowered in malnutrition. For example, neutrophil activity is reduced, interferon response is decreased, and complement 3 and factor B of the complement system are decreased in protein-calorie malnutrition. Also, deficiency of vitamin A, vitamin C, and folic acid makes an individual highly susceptible to infection by many microbial pathogens. Concerning hormonal level, it's important for you to know that individuals with certain hormonal disorders become increasingly susceptible to infection. For example, people suffering from diabetes mellitus, hypothyroidism, and adrenal dysfunction are highly susceptible to staphylococcal, streptococcal, candidiasis, aspergillosis, among other microbial infections. Similarly, pregnant women are more susceptible to many infections due to higher level of steroid hormone during pregnancy. So you also need to remember that the innate immunity performs two major functions. Number one, it kills disease-causing microorganisms that have invaded the host. And number two, it also activates the adaptive immune system. So the second type of immunity is adaptive, which is also referred to as the acquired immunity. And this is the mechanism that are stimulated by microbes or the disease-causing agents and are capable of recognizing both microbial and non-microbial substances. And they are also referred to as acquired immunity. So the distinguishing features of the adaptive immunity include: number one, it is highly specific. Number two, it requires several days before becoming effective, and it improves after exposure, and it also has immunological memory. And we have two types of acquired immunity, that is the active immunity and the passive immunity. But it is also very critical for you to know that acquired immunity can be induced by two things. Number one, it can be induced by the host's response to the disease-causing agent, and it can also be induced as a result of transfer of antibodies or lymphocytes specific for the microorganisms. So we are going to discuss the two types of acquired immunity. We will start with active immunity. So active immunity is the resistance that develops by an individual after contact with foreign antigens or disease-causing microorganisms. And contact can be in various forms. For example, it can be in form of an infection, which can either be clinical or subclinical, and this will form the natural active immunity. Second, contact can also be as a result of immunization with live or weakened or killed or attenuated infectious agents or their antigens, and this will form the artificial active immunity. And again, contact can also be as a result of exposure of microbial products, for example, toxins and toxoids. And you also need to remember that the host actively produces an immune response as a result of those contacts. Now, what are the advantages and the disadvantages of active immunity? So the advantage is that it provides long-term resistance, whereas for the disadvantage is that there is slow onset. So active immunity is mediated by two things. Number one, it is mediated by the humoral immunity, and number two, it is mediated by the cell-mediated immunity. So we are going to discuss this briefly one by one. So starting with cell-mediated immunity, you need to understand that this type of immunity is mediated by cells, and it protects the host against fungi, viruses, and facultative intracellular bacterial pathogens. And cell-mediated immunity mediates delayed hypersensitivity, that is type 4 hypersensitivity. And it is only T cell dependent antigens that leads to cell-mediated immunity. You also need to know that both CD4 and CD8 T cells are involved in cell-mediated immunity. And this type of immunity provides immunological surveillance and immunity against cancer. And it also participates in rejection of homografts and graft versus host reactions. Moving on to humoral immunity. So this type of immunity is mediated by the antibodies which are produced by the B cells, and it protects the host against intracellular bacterial pathogens and viruses infecting respiratory or intestinal tract, and it also prevents recurrence of the microorganisms. So it is a type of immunity that mediates immediate hypersensitivity, that is for type 1, type 2, and type 3 hypersensitivity. And you also need to know that in this type of immunity, B cells directly bind soluble antigens, resulting in production of the antibodies. And it is only T helper cells that are involved in humoral immunity. And for humoral immunity, it has got no major role in immunological surveillance, but may be involved in early graft rejection due to preformed antibodies. So I have given here a table that gives the comparison of cell-mediated immunity and humoral immunity. You can find time and check on that. So next we move to passive immunity. So passive immunity is the resistance that is based on antibodies that are preformed in another host. And it can either be artificial or natural. So examples of artificial passive immunity includes administration of antibodies against diphtheria, tetanus, and botulism, among other diseases. And it can also include administration of preformed antibodies to certain viruses, for example, the rabies virus, hepatitis A virus, and hepatitis B virus. Examples of natural passive immunity will include passage of immunoglobulin G from the mother to the fetus during pregnancy, and also passage of immunoglobulin A from the mother to the newborn during breastfeeding. Now, what are the advantages and the disadvantages of passive immunity? So for the advantage of passive immunity is that there is prompt availability of large amounts of antibodies. And for the disadvantages, the antibodies have a short lifespan, and number two, there is possibility of hypersensitivity reaction, particularly if the antibodies are from another species. So characteristics of passive and active immunity, I have summarized it in form of a table here, and we are going to highlight it briefly. So we shall discuss this in respect to the mediators, their advantages and their disadvantages. So active immunity is mediated by antibodies and T cells, whereas passive immunity is mediated by only antibodies. The advantage of active immunity is that it provides long-term resistance, whereas the advantage of passive immunity is that there is prompt availability of the antibodies. Then the disadvantage of active immunity is that there is slow onset, and the disadvantage of passive immunity is that there is short duration. It acts within months only. So next we are going to discuss passive active immunity. So this involves giving both preformed antibodies, which will help in providing immediate protection, and this is achieved by passive immunity. And also it also involves providing long-term protection by the use of active immunity. So the preparations which will induce both passive and active immunity must be given at different body sites to prevent neutralization of immunogens in the vaccine by the given antibodies. For example, this type of combination is seen in the prevention of tetanus, rabies, and hepatitis B. So you also need to know the main components of innate and adaptive immunity. And like I pointed out, innate and adaptive immunity have two arms, the humoral arm and the cell-mediated arm. So we are going to discuss the major components of the innate and adaptive immunity under the humoral arm and under the cell-mediated arm. So the humoral immunity of the innate immunity is mainly made up of the complement and neutrophils, whereas the humoral immunity of the adaptive immunity is composed of the B lymphocytes and the antibodies, which are made from the plasma cells. Next, the cell-mediated immunity of the innate immunity is composed of the macrophages and dendritic cells. And remember, macrophages and dendritic cells are antigen-presenting cells. We are going to discuss them briefly. And for the adaptive immunity, the cell-mediated component is going to be mediated by helper T cells and cytotoxic T cells. So this table is very important for your exams. You need to take note of it. And there is also a very important point that you need to remember for your exams, and that is macrophages and other antigen presenting cells, for example dendritic cells, have a role in both innate and adaptive arm of the immunity, and I'm going to explain how this is the case. Considering phagocytosis of microorganisms by macrophages and dendritic cells, this process can result into, number one, destruction of the microbes and secretion of cytokines which fall under the innate immunity arm. It can also act as antigen presenting cells. Understand, I've said macrophages and dendritic cells are antigen presenting cells, so they can present the antigens to CD4 positive helper T cell, and that falls under the adaptive arm of the immune system. So next, we are going to discuss important features of innate and adaptive immunity. So regarding specificity, innate immunity is non-specific, whereas adaptive immunity is highly specific. Regarding the time required for effect after exposure, innate immunity is going to act within minutes, whereas adaptive immunity requires several days for effect after exposure. Regarding whether it improves after exposure or no, for the innate immunity, it does not improve after exposure, whereas for adaptive immunity, it improves after exposure. Regarding memory, innate immunity have no immunological memory, whereas adaptive immunity has immunological memory. So next, we are going to discuss the interaction between innate and adaptive immunity. And to explain that, I've drawn a diagram on the whiteboard. So from the diagram on the left side, we have the major components of the humoral immunity. And on the right side, we have the major components of the cell-mediated immunity. So it is important for you to know that humoral immunity is our main defense mechanism against extracellular, encapsulated, and pyrogenic bacteria. That is very important for you to remember for your exams. Now, with the help of interleukin 4 and interleukin 5, which are derived from helper T cell, the B lymphocyte is going to be transformed into a plasma cell. So we can see that transformation there of a B lymphocyte to a plasma cell. And the plasma cells are going to produce antibodies which can neutralize toxins and viruses. So antibodies are produced from the plasma cells after the B lymphocytes have undergone transformation under the influence or under the stimulation of interleukin 4 and interleukin 5, which are produced by helper T cell. Now, what about other components of the humoral? So the other components of the humoral immunity, which includes the complement and the neutrophils, act by killing bacteria and other offending agents. Now, on the right side, like I've already said, we have the major components of the cell-mediated immunity. We have helper, we have the T cell, which gives us a helper T cell and cytotoxic T cell. So activated helper T cell is going to inhibit intracellular bacteria and fungi, whereas activated cytotoxic T lymphocyte will kill virally infected cell. So next we will discuss important components of innate immunity, and this one we are going to discuss it under two subtopics. Number one, factors that limit entry of microorganisms into our body, And number 2, factors that limit growth of microorganisms inside our body. So we shall start with factors that limit entry of microorganisms into our body. So we shall be looking at the factor and its or their respective modes of action. So we'll start with keratin layer of the intact skin. So this forms part of the anatomical barrier, and they provide mechanical barrier to entry of disease-causing agents into the host body. For example, an intact skin prevents entry of microorganisms into the body. So any breaks in the skin due to scratches, wounds, or abrasion will cause infection because it will allow for the entry of the microorganisms into our body. Bites of insects harboring pathogenic organisms can also introduce the pathogens into the body and transmit the infection. You also need to remember that skin secretes sebum, which prevents growth of many microorganisms. And this is because sebum consists of lactic acid and fatty acids that maintain the pH of the skin within the range of 3 to 5. And this pH range inhibits growth of most microorganisms. We also have another factor that provides mechanical barrier, and that is the mucous membrane. So mucous membranes form a large part of outer covering of gastrointestinal, respiratory, genitourinary, and many other tracts of the human host. And from the mucous membrane, we can obtain secretions, for example, saliva, tears, and mucus secretions. So what is the importance of these secretions in regard to limiting the entry of microorganisms into our body? So these secretions can wash away potential invading microorganisms, preventing their attachment to the initial state of the infection. It can also contain, or some of them contain, antibacterial or antiviral substances that kill pathogens. And also, you need to remember that mucus secreted by the epithelial cells of the mucous membrane entraps invading microorganisms. Then in the lower respiratory tract, mucous membrane is covered by cilia, and the movement of cilia propels mucus entrapped microorganisms from the tracts. So the other factor is low pH in the stomach, vagina, and fatty acids in the skin. So these ones are going to retard growth of microorganisms. And it is important for you to know that very few ingested microorganisms, for example, can survive the low pH of the stomach content. So the next factor is lysozyme in tears and other secretions. And what these do, they degrade peptidoglycan in bacterial cell wall. The next factor is defensins, or sometimes we refer to it as cationic peptide. So these ones, what they do, they create pores in the membrane of microorganisms. Then we have the normal flora of the throat, colon, and the vagina. So these ones, they occupy receptors preventing colonization. So the normal flora basically compete with the pathogens for attachment sites on the epithelial cell surface and for necessary nutrients, and as a result of that, then they minimize or prevent colonization of the disease-causing microbes. So next, we shall look at factors that limit growth of microorganisms inside our body. So the first factor is neutrophils, and neutrophils ingest and destroy microorganisms, of course, by the process of phagocytosis. So maybe just to have an overview, phagocytosis is a process of ingestion of extracellular particulate material by certain specialized cells. For example, in this case, we are talking of the neutrophils. We can also talk of tissue macrophages and even the monocytes. So Phagocytosis is just a type of endocytosis in which invading microorganisms present in the environment are ingested by the phagocytic cells. And it is also important for you to know that in this process, plasma membrane of the cell expands around the particulate material, which may include whole pathogenic microorganisms, to form large vesicles called phagosomes. So the next factor is natural killer cells. What they do, so these ones kill virally infected cells. Then next we have macrophages and dendritic cells. So these ones ingest and destroy microbes, and they also act as antigen presenting cells. Then we have interferons. These ones inhibit viral replication. Then we have complement system. For example, complement 3b acts as opsonin, and they can also attack the membrane complex, creating holes in the bacterial membrane. The next we have transferrin and lactoferrin. So these ones, they sequester iron required for growth of bacteria, and inflammatory responses limit spread of microbes. One caution that you need to understand: tissue damage caused by a wound or an invading pathogenic microorganism induced a complex sequence of events collectively known as inflammatory response. And the end result of inflammation may be, number one, the activation of the specific immune response to the invading agent, or number two, it can also clear the invader by the components of the innate immune system. Fever is another factor. So these ones elevate temperature to retard microbial growth. And lastly, we have a factor called apolipoprotein B RNA editing enzyme. And what these ones do, they cause hypermutation in the retroviral DNA and mRNA. So moving on, next we have to discuss components of the innate immunity. So we have the epithelia as one of the components, and this one provides mechanical barrier to the entry of microorganisms from the external environment, and they also produce defenses. So lymphocytes located in the epithelia combat microorganisms at these sites. The next we have monocytes and neutrophils. Like we have seen, those two, they participate in phagocytosis, so they are phagocytes in blood, so can be rapidly recruited to the site of infection. And monocytes that enter the tissues and mature are referred to as macrophages. So basically macrophages are matured monocytes. Next, we have the dendritic cells, which I've said are examples of antigen presenting cells. So these are specialized population of cells that are found in the epithelia, the lymphoid organs, and also in most of the tissues. So they present the antigens, or you can talk of the viral peptides for recognition by the T lymphocytes. So they have rich collection of receptors that can sense microorganisms, damaged cells, and stimulate secretion of cytokines. Then for the natural killer cells, these ones prevent or provide early protection against viruses and intracellular bacteria. Then for the innate lymphoid cells, these are cells that appear like lymphocytes, but they have got some features like cells of the innate immunity. Then we have three types of soluble proteins: the complement system, mannose binding lectin, and C-reactive protein. We shall be discussing them in the course of our subsequent presentations. And lastly, we have the lung surfactant. So regarding components of the adaptive immunity, you need to know that this one is made up of the lymphocytes and their products, including the antibodies, and they are highly diverse because they have got diverse receptors that are used to recognize a vast array of foreign substances. Then we have to look at the cells of the adaptive immunity, and they are made up of, this is made up of two cells, the T and the B lymphocytes. So T and B lymphocytes, they appear different, and they are specialized in molecular properties and their respective functions. So lymphocytes and other cells involved in the immune response, they are constantly circulating among the lymphoid and other tissues via blood and lymphatic circulation. And since they are constantly circulating within the body system, then they promote immunological surveillance. Then naive lymphocytes, these are mature lymphocytes that have not encountered the specific antigen for which they have the specific receptors, and they are also referred to as immunologically inexperienced lymphocytes. So when naive lymphocytes undergo activation by recognition of specific antigens and other signals, then they differentiate into effector cells and memory cells. So we get effector cells and memory cells from differentiation of naive lymphocytes once they have undergone activation. Now, for effector cells, they perform the function of eliminating the microorganism, whereas the memory cells, they live in a state of rest awareness. Then in case of re-entry of the microbe into the host, then the memory cells can react rapidly and strongly to combat the microorganism. Then we have three populations of T lymphocytes: the helper T lymphocytes, cytotoxic T lymphocytes, and regulatory T lymphocytes. So helper T cells, they stimulate the B lymphocytes to produce the antibodies, and they can also activate other leukocytes, for example phagocytes, to destroy microorganisms. And for cytotoxic T lymphocytes, these ones kill virally infected cells, whereas for the regulatory T lymphocytes, they limit the immune responses, and they also prevent the reaction against self-antigen, that is autoimmune reactions. Then next we have to discuss commonly used terms in immunology, and one of the term is antigen. So antigens, basically these are molecules that react with antibodies, and we can also define an antigen as a substance which stimulates antibody production when introduced into the tissues, and they're usually protein in nature. Immunogens are molecules that induce an immune response, and in most cases antigens are immunogens, and that's why the two terms are used interchangeably, except for the haptens. So what are haptens? So a hapten is just an unprotein substance which has no immunologic property by itself, but on combining with a protein, it can form a new antigen capable of producing antibodies. Then antibodies, these are just protein substances that are produced as a result of antigenic stimulation. So circulating antibodies are referred to as immunoglobulins, and there are 5 of them: immunoglobulin A, G, M, E, and D. You can remember them using the acronym GAMED, and we are going to discuss these 5 classes of immunoglobulins in our subsequent presentation. Then lastly, we will discuss briefly on the structure of the immune system. So the immune system is composed of 2 structures, that is the primary lymphoid organs and the secondary lymphoid organs. So the primary lymphoid organs consists of the thymus and the bone marrow, whereas for the secondary lymphoid organs consist of the lymph node, the spleen, and mucosa associated lymphoid tissue in the respiratory and gastrointestinal tract. So this concludes our first presentation. I hope this presentation is helpful, and for you to understand and know more, I would recommend you check in your textbooks. So if you like my presentation, please share and also comment on my YouTube page. Until next time, keep safe and be blessed. Thank you.